Good day, this is Professor Will, CBMD, PhD. Today is Saturday, March 23rd, 2019. It is 14:11:59 seconds Eastern Standard Time. And today I'm going to be doing a study on amoebas. So let's get started here. Amoeba cells grow larger than bacteria, but the continually changing shape makes size measurements difficult. Amoeba can range from about 40 micrometers (UM) to more than 600 micrometers in diameter. Their ever-changing structure makes amoeba equally difficult to identify the species or even genus level under a microscope. As bacteria do, amoeba produce asexually by splitting into a process called binary fission. As all other eukaryotic cells do, amoeba have distinct membrane-enclosed organelles such as a nucleus. Some species have many nuclei, mitochondria, water-containing contractile vacuoles, and food vessels in their cytoplasm. Amoeba are also divided into two general groups based on morphology, cell structure: naked amoeba and shelled amoeba. Naked amoeba possesses a flexible membrane that holds in the cell contents. Shelled amoeba contain an outer structure called a shell or a test, which provides the cell with a characteristic shape. The shell is rather fragile, however does not provide the physical protection and strength seen in other protozoa that make much stronger forms called cysts. Many shelled amoeba travel by pseudopodia, by extending a pseudopod out of their shell and then moving in that direction. Amoeba possesses methods of mobility without the use of pseudopods. For example, amoeba move by changing the consistency of their cytoplasm. Cytoplasm is the aqueous material inside all prokaryotic and eukaryotic cells. The eukaryote cytoplasm occurs inside the membrane but outside the nucleus and other organelles. Amoeba change their cytoplasm depending on their need to move. When they prepare to become motile, their cytoplasm becomes soft and fluid and is referred to as plasmasol. Plasmasol gives the cell flexibility. When the cell stops moving, the cytoplasm becomes slightly firmer and is called plasmagel. Microbiologists have not fully explained how plasmasol turns into plasmagel and back. It is known, however, that high pressure on the outside of the cell hastens plasmagels break down to fluid, and the compound hyaline gives the gel its firmness, especially inside pseudopods. Because they are found only in aqueous environments, amoeba are good at floating with currents as another means of motility. To do this efficiently, amoeba cells extend several pseudopods in different directions at once to create a star-like shape. Under the microscope or a video, amoeba appears rather sluggish when they move through water. Amoeba eat by engulfing their prey, which may include small bits of organic matter, bacteria, algae, or other protozoa. They have no trouble catching non-motile algae and bacteria by extending pseudopods around them and engulfing them. But ciliated protozoa and flagellated bacteria contain many short cilia or long flagella, respectively, which make them fast swimmers. Amoeba must move at least as fast to catch these cells. Amoeba extend their cell bodies entirely around their food or prey before actually touching it. Once completely surrounding the item, the amoeba cell gathers it into the cytoplasm in a process called pinocytosis. Pinocytosis is similar to phagocytosis that occurs in the mammalian bloodstream, in which white blood cells envelope foreign material in the blood. The amoeba draws the food into a vacuole into which it releases digestive enzymes. The enzymes degrade the food into small pieces that the cell can then absorb into its cytoplasm. Amoeba, meanwhile, excrete waste and carbon dioxide through their cell membrane and take in oxygen through the membrane. They carry out oxygen and carbon dioxide exchange by simple diffusion. Simple diffusion is a process that does not cost the cell any energy to perform because materials cross the membrane without the need for active transport systems. By contrast, waste excretion requires energy. Amoeboids is a term for microbial species that are not true amoeba, but move as an amoeba do by using pseudopods. This type of motility is called amoeboid movement. Examples of organisms that use amoeboid movement are slime molds, water molds, members of the protozoal subphylum Sarcodina, and some algae, chlorokinophytes. Amoeba habitats. Amoeba's fragile cell structure forces them to inhabit moist environments. When amoeba live in soils rather than water, they must inhabit only soils with high moisture content. The digestive tract of humans and other animals provides an aqueous environment where amoeba exist and can cause potential health problems. The vast majority of amoeba, however, live as harmless inhabitants of the environment in ponds, lakes, and other natural bodies of water. Aquatic amoeba can live in either freshwater or saltwater habitats. Freshwater habitats include ponds, ditches, and slow-moving waters high in organic material. Some amoeba live in marine habitats, but most saltwater amoeba prefer brackish, salty, but not as salty as the ocean waters rather than ocean waters. Amoeba have certain requirements for the type of water in which they live due to their need for isotonic conditions. Isotonic conditions describes a situation in which the pressure outside a cell equals the pressure inside the cell. Unlike bacteria, amoeba do not have a rigid cell wall to protect them from too much or too little pressure in their surroundings. Most cells rely on the process of osmosis to regulate pressure. Osmosis is a transfer of water into or out of the cell to keep the pressure of the contents equal to the pressure in the environment. But because amoeba do not have this ability and also lack a cell wall, both hypertonic and hypotonic conditions can injure or kill amoeba. In hypertonic conditions, the concentration of matter outside the cell is greater than that inside. When this occurs, water rushes out of the cell and it shrivels. In hypertonic conditions, the concentration of matter outside the cell is less than that inside. Water then rushes into the cell and then eventually bursts. Amoeba in nature seek habitats that provide the correct osmotic conditions for keeping them alive. Microbiologists who study amoeba in a laboratory specialty called protozoology ensure that the osmotic pressure is safe for the cells being studied. For this purpose, laboratory media usually contains 7.5 grams of salt, NaCl, along with other nutrients per liter of water to provide osmotic conditions favorable to amoeba cells. Good day, this is Professor Will, CBMD, PhD, today Saturday, March 23rd, 2019, and this concludes my brief description on amoebas. Hope you enjoyed listening. Have a great day.